I'm Dr. Abhinav Singh. I'm a postdoc in Mark Humphrey's group, the Systems Neurophysiology Lab at the University of Manchester. Our lab is primarily interested in the problem of neural coding, specifically population coding. The problem, simply stated, is that our brain makes a transformation about the information about the external world, which comes in the form of sensory signals such as light, sound, into an internal representation of the world which happens in the form of spikes. How this transformation happens is currently unknown. And we are very interested in trying to make progress on that problem. Specifically, we are currently interested in understanding how spikes in the prefrontal cortex actually encode information about rule learning, and specifically how rule learning transforms the statistical structure of spiking inside the prefrontal cortex, and that's what we are trying to disentangle right now. So our lab is primarily computational, so we of course collaborate with experimentalists who of course mainly use electrophysiological recordings. Our main tools probably are sort of a desktop computer and occasionally use of a clusters, but mostly MATLAB and Python probably are, and of course pencil and paper are our main tools that we use in our lab. Oh, this trial, that was kind of fantastic thing. It was probably one of the first ways of actually seeing probably the nervous system in a 3D form and in an interactive fashion, which is quite unprecedented. Of course, everybody's seen, all neuroscientists have seen images of neurons or of axons, but it's quite a different experience of actually seeing an axon or a neuron from the inside. And I would imagine that it has a lot of potential in exploratory analysis, because it really allows you to just roam around the brain, which is kind of an interesting experience, which I've certainly never had before, and hopefully many people will have tomorrow. Right now, there are two key benefits. I would imagine it has tremendous educational benefits, because it would really give a very realistic idea of what the structure of a nervous system looks like for students, as well as probably it offers an interesting exploratory opportunity to just roam around the cell, the brain structure, to see whether an interesting pattern comes up. So as an exploratory tool, to see unexpected patterns which might only become visible when you actually roam around inside that, it might turn out to be an interesting tool in the future. So my future goal is to understand how the sensory world is being transformed into neural spikes. This will advance our fundamental understanding of the nervous system at the systems level, but will also have numerous clinical applications in treating patients with sensory disorders, as well as perhaps in future applications in robotics to make better sensors.